Welcome everyone. Today I will discuss about culture media. Culture media is artificial food. Artificial food for cultivation of bacteria in the laboratory. Culture media is artificial food for cultivation of bacteria in the laboratory. Now come to classification of culture media. On the basis of consistency, culture media is three types: solid, semi-solid, and liquid media. Solid, semi-solid and liquid media. Solid media are blood agar media, chocolate agar media, McConaughey agar media. Mainly these agar containing media are solid media. Agar is a solidifying agent and it is called most ideal solidifying agent because it is biologically inert and it is also transparent. That's why the colony morphology can be observed very well and it is transparent. It remains solid at room temperature and doesn't add any nutritive value or doesn't change pH of the media. That's why this agar is called ideal solidifying agent. Agar is derived from seaweed and it is a polysaccharide, that is polymer of D-galactose pyranose. Agar is polymer of D-galactose pyranose. An example of semi-solid media is MIU media. MIU means motility, indole, urea agar media. Then liquid media. Liquid media are peptide, sugar broth, bile broth. Usually the word broth is added with name of liquid media. Sugar broth, bile broth, these are example of liquid media. Then on the basis of presence of oxygen, culture media is two types. Number one, aerobic media. Number two, anaerobic media. In aerobic media, bacteria is grown in presence of oxygen. And in anaerobic media, bacteria grows in absence of oxygen. Most of the media are aerobic media, and example of anaerobic media are Robertson cooked meat media, thioglycolate broth. In Robertson cooked meat media, Minced beef is used. Minced beef. Then come to most important classification according to chemical or nutritional composition. According to chemical and nutritional composition, culture media is mainly two types: routine laboratory media and synthetic media. Routine laboratory media and synthetic media. Synthetic media is again two types: simple and complex media. And routine laboratory media is again two types: basal media and special media. Example of basal media are peptone water, nutrient broth. From basal media, we can prepare other media by adding special substances. That's why this media is called basal media. And special media are transport media. Special media are transport media, indicator media, enriched media, selective media, and storage media. Special media is of several types like transport media, indicator media, enriched media, selective media, and storage media. Now I will describe transport media, indicator media, enriched media, selective media, storage media one by one. First come to transport media. This transport media are used to carry a specimen containing microorganism from site of collection of the specimen to the laboratory. When there is delay in testing the specimen or there is a long distance between collection site and the laboratory, then transport media are used to carry the specimen. This transport media keep desired bacteria viable and inhibit growth of unwanted bacteria. So the purpose of this transport media is to keep desired bacteria viable and to inhibit unwanted bacterial growth or to inhibit growth of contaminating bacteria. Now, example of transport media. MI is transport media. Stewart media. These two media are used to carry Gonokokkai. MI is transport media and Stewart media. These two media are used for transport of Gonokokkai. Monsource media, alkaline peptonoter. These two media are used for carrying Vibrio cholerae. And last one, Carry-Blair media. Carry. This is not C-A-R-R. Here, single R. C-A-R-Y. Carry-Blair media. This media is used for transporting Salmonella and Shigella. Amy's and Stewart media for Gonococci, Monsource media, alkaline peptonoter for Vibrio cholerae, and Carry-Blair media for Salmonella Shigella. Then enriched media. These media are enriched with some special substances like blood, serum, egg to support the growth of desired bacteria. So these enriched media are enriched with blood, egg, serum to support growth of some desired bacteria. An example of enriched media, number 1, blood agar media. Blood agar media. In blood agar media, it is enriched by 5% sheep blood. 5% sheep blood is used in blood agar media. So, for preparation of 100 ml blood agar media, 95 ml blood agar base is mixed with 5 ml sheep blood. That's why it is 5% sheep blood. That means in 100 ml of blood agar media, 5 ml will be sheep blood. And here we don't usually use human blood because in human blood there may be antibiotic and there may be some heavy metal which inhibit growth of microorganism. Then chocolate agar media is another enriched media. It is not enriched with chocolate. Basically, this chocolate agar media is named from the color of the media. Color of the media. And this chocolate agar media is heated blood agar media. When blood agar media is heated at 80 degrees Celsius, then the RBC are lysed, hemoglobin comes out from the RBC and the color of the media changes to chocolate brown. Then it is called chocolate agar media. In blood agar media, both gram positive and gram negative bacteria grows. And in chocolate agar media, some fastidious bacteria grows like Streptococcus pneumonia, Haemophilus influenzae, and Neisseria species. Neisseria species. So example of enriched media, number 1, blood agar media, where both gram positive and gram negative bacteria grows. Then chocolate agar media, here pneumococci, Haemophilus influenzae, Neisseria species grows. And number 3 is Lowenstein-Jensen media. This media is enriched by egg and used for culture of Mycobacterium tuberculosis. Lowenstein-Jensen media is enriched with egg and used for cultivation of Mycobacterium tuberculosis. Now come to selective media. This selective media enhance growth of some specific bacteria and inhibit growth of other bacteria. Enhance growth of specific bacteria and inhibit growth of other bacteria. That's why this is called selective media. Now come to example of selective media. We have to know the name of media, selective ingredients, and bacteria grows here. Number 1, McConkey agar media. This McConkey agar media is selective for Enterobacteriaceae. Enterobacteriaceae is a large family, and in future we will learn about Enterobacteriaceae. This McConkey agar media is selective for Enterobacteriaceae, and it is made selective by adding bile salt and crystal violet. McConkey agar media selective for Enterobacteriaceae, and it is made selective by adding bile salt and crystal violet. Then number 2, Lowenstein-Jensen media. This media is selective for Mycobacterium tuberculosis, and this media is made selective by adding malachite green. Lowenstein-Jensen media selective for Mycobacterium tuberculosis and made selective by adding malachite green. Then number 3, Monsource media. Another name is telluride turoculate gelatin agar media. Monsource media or telluride turoculate gelatin agar media. This Monsource media is selective for Vibrio cholerae. Monsource media is selective for Vibrio cholerae, and it is made selective by adding bile salt and potassium tellurite. Monsource media, another name is telluride tellurium, telluride, turoculate, gelatin, agar media. Telluride, turoculate, gelatin, agar media. This media is selective for Vibrio cholerae and made selective by adding bile salt and potassium telluride. So, media name is telluride, turoculate, gelatin, agar media. And number 4, blood telluride agar media. This media is selective for Corynebacterium diphtheria. Corynebacterium diphtheria. And this media is made selective for Corynebacterium diphtheria by adding potassium telluride. So these are the example of selective media. First, McConkie agar media, Lowenstein-Jensen media, Monsour's media, or telluride, turoculate, gelatin agar media, and last, blood telluride agar media. This media is selective for Corynebacterium diphtheria and made selective by adding potassium telluride. Then come to indicator media. In indicator media, some indicators are used. Those indicators indicate some specific bacteria by making any color change or change in any other form in the media. Now, examples of indicator media. We have to learn name of media, indicator used, and bacteria grow in that media. Number 1 is McConkie agar media. In this media, we use lactose and neutral red as indicator, and here Enterobacteriaceae bacteria grows. We found two types of colonies in McConkie agar media. Some are lactose fermenting and some are non-lactose fermenting. Lactose fermenting bacteria produce pink colony and non-lactose fermenting bacteria produce pale colony. Lactose fermenting bacteria produce pink colony because after lactose fermentation there will be production of lactic acid. This lactic acid decrease the pH of the media. So this indicator neutral red produce acidic color, that is red or pink. That's why lactose fermenting bacteria produce pink color colony on McConkie agar media. After lactose fermentation, there will be production of lactic acid, decrease in pH, and this neutral red indicator shows acidic pink color. Then blood agar media. Blood agar media is both indicator media and also enriched media. In blood agar media, indicator is hemolysis of RBC, and bacteria grows is Streptococcus. In blood agar media, we found three types of hemolysis: alpha hemolytic streptococci, beta hemolytic streptococci, and gamma hemolytic streptococci. Alpha hemolysis means incomplete hemolysis of RBC. Incomplete hemolysis of RBC. Beta hemolysis means complete hemolysis of RBC. And gamma hemolysis is a misnomer. Actually, there is no hemolysis. So, in case of blood agar media, the indicator is hemolysis, and we can differentiate alpha hemolytic, beta hemolytic, and gamma hemolytic streptococci from after seeing the pattern of hemolysis. Then number 3 is Monsource media or telluride, turocolate, gelatin agar media. Here indicator is potassium telluride. Indicator is potassium telluride, and bacteria grows here is Vibrio cholerae. This potassium telluride produces blackening around the colony. Blackening of the colony is due to this potassium telluride. And number 4 is blood telluride agar media. Here also the indicator is potassium telluride, and bacteria grows is Corynebacterium diphtheria. So in blood telluride agar media, this potassium telluride indicator produce grayish black colony of Corynebacterium diphtheria. And this is end of culture media from today. Hope this will be helpful for you, and next day I will try to discuss some other topic. Thank you for staying with me. Thank you everyone.